Good evening, postgraduate, and welcome to this journal club from All India Ophthalmological Society Academic and Research Committee. Well, this is a PG program. We call it with a difference because unlike other postgraduate teaching programs, we tend here to have topics which are not routinely available to you in other PG programs. So journal club is the one where we are seeing in extinction in majority of the institutes in our country, and that is the reason we wanted to have it at this particular forum. So in today's journal club we will be having two presentations. Two articles would be presented. The first article would be presented from Shankar Nethralaya, Kolkata, by Dr. Rohan Basak, who is a comprehensive ophthalmology fellow, and his mentor is going to be Dr. Monideepa Banerjee, who is a consultant in Department of Pediatric Ophthalmology and Neuro-Ophthalmology at Shankar Nethralaya, Kolkata. So, without much ado, can I request Rohan to share his screen? Dr. Tinku Bali, our member ARC North, is going to be the moderator for today's program, as she is slightly delayed because of the rains. Till then, I will be carrying forward the program.
Is the screen visible for everyone?
Yeah, Dr. Rohan. Yeah, before I just allow you, let me also thank all the senior discussant, Dr. Parvez Khan, who is a professor at Department of Ophthalmology, GSVM Medical College, Kanpur. Professor Namrata Sharma, who is Professor of Ophthalmology at RP Center, Delhi. And Dr. Shweta Agrawal, who is Senior Consultant of Cornea and Ocular Surfaces from Shankar Nethralaya, Chennai. Of course, I will be thanking Dr. Rohit Shetty, who is also is going to mentor one of the articles today. Okay, over to you, Rohan, please.
Thank you, sir. So, good evening, everyone. I'm Dr. Rohan from Shankar Nethralaya, and with my mentor, Dr. Monideepa Banerjee, I'll be delving into various innovations in regenerative ophthalmology. At the outset, I'd like to thank Team AIOS and Sarakshi Nethralaya for this wonderful opportunity. The human body has a remarkable property of regeneration facilitated by stem cells. They were first conceptualized by German scientist Ernst Haeckel in 1868 as unicellular ancestors, and later coined by Boveri and Hacker. These cells exhibit three defining characteristics. Firstly, they can undergo numerous cell cycles or self-renewal. Secondly, they originate from a single progenitor cell. And lastly, they have the potential to differentiate into specialized cellular phenotypes. Based on the potency, they can be of various types. The TOTI or omnipotent stem cells are present only during early embryogenesis and give rise to both embryonic and extraembryonic tissues. They have no current clinical use. In contrast, the pluripotent stem cells generate all three germ layers, the mesoderm, surface, and neuroectoderm. They have they have immense role in ophthalmology. And they also have no extraembryonic role. Now that you have heard of the TOTI and pluripotent cells, let's move on to multipotent stem cells. They differ from the previous two as they create multiple lineages but within a single germ cell layer. A classic example would be the neural crest cells forming most of the cornea and ciliary muscles. The other types of stem cells are the oligopotent stem cells, widely applied in clinical practice. They produce two or more lineages within a specific tissue, just like the limbal epithelial stem cells forming both corneal and the conjunctival epithelium. Finally, unipotent stem cells can give rise to a single cell type within their lineage. Unfortunately, not all the 5 types of stem cells can be utilized in ophthalmology. In most anterior segment pathologies, the somatic or adult stem cells, which are either multi or pluripotent, are invaluable. They are found in various niches among multiple organs, like the bone marrow, the adipose tissue, among others. On the other hand, posterior segment diseases warrant the use of more pluripotent cells, like the embryonic stem cells from the blastocyst and umbilical cord, or modified somatic cells called the induced pluripotent stem cells, or the iPSCs. Due to their sourcing and proliferative potential, they face the risk of neoplasia and ethical concern. With this background, Let us start the discussion on advancements in ocular regenerative therapies. This is a review article published by a group of Polish researchers, Tomczak et al., in a high-impact journal, Biology, in 2023. There has been a recent shift in the publications pertaining to stem cells. Their numbers in 2021 exceeded the cumulative total until the end of 2007, according to the PubMed database. So, is ocular regenerative therapy only limited to stem cells? Not quite. It is a long-term therapy aimed at replacing dysfunctional cells and restoring existing stem cells by the utilization of natural or engineered biological tissues, extracts, or materials. It encompasses stem cell therapy, gene therapy, and tissue engineering. The ocular regenerative therapy is not just restricted to the ocular coats, but also finds place in periocular structures such as the conjunctiva, lacrimal gland, orbital fat, hair follicles, and meibomian glands as well. Cornea is the most important structure for most regenerative therapies because of its abundance of stem cells. In the cornea, the limbal epithelial stem cells housed in the vas palisades play a pivotal role. The stromal and endothelial stem cells, unlike their limbal counterparts, exhibit limited regenerative capacity. The choice of stem cells depends on the laterality and the severity of the disease. Broadly, epithelial transplants may be autografts, allografts, or non-limbal grafts. In case of unilateral conditions, we opt for techniques utilizing the patient's own tissue. One such technique is the conjunctival limbal autograft, or CLAV. This involves the transplantation of two large grafts from the contralateral healthy eye. Despite favorable outcomes, it leads to an iatrogenic stem cell deficiency in the donor side because of a large chunk of tissue removed. To counter this, newer techniques like the mini-CLAV employ smaller grafts. For cases with insufficient donor tissue, another limbal technique called the cultivated limbal epithelial transplantation, or CLET, was introduced. Compared to CLAV, this staged procedure used lab-cultivated autologous or allogenic tissues. Here, smaller grafts are taken in a microcentrifuge tube and harvested onto various matrices. Even with promising results, it was tedious, time-consuming, and did not simulate in vivo conditions. To further reduce the graft size and immunogenic load, Sangwan et al. From India introduced a novel technique. This involved dividing the graft into smaller bits and fixing them circumferentially in the peripheral cornea using amniotic membrane, glue, and bandage contact lens. They named this simple method of transplanting the limbal epithelial stem cells as SLET. Though comparable to both CLAV and CLET, there was a risk of graft extrusion. This issue was addressed by modifications like the MSLET that used an additional amniotic membrane or fibrin glue layer, thus improving the stability. Bilateral conditions, however, cause a dearth of viable stem cells. In such cases, we turn to other limbal sources such as cadaveric keratolimbal allografts. Conjunctival limbal tissue from live-related donors may also be used, as shown in the picture above. As they are foreign tissues, they need to be screened for transmissible diseases. Both approaches fare poorer than autografting and necessitate chronic immunosuppression. The picture below shows a combination of auto and allografts to reduce the immunogenic load. For more extensive diseases, the limbal tissue just does not suffice. This led to the exploration of alternate sources, and one such approach is COMET, pioneered in Japan, which employs a buccal mucosal graft. The procedure resembles CLET, but uses a carrier-free epithelial sheet mounted on a donut-shaped supporter and later transferred on to the stromal bed. It is now a regulatory-approved therapy marketed worldwide. Although less immunogenic with good outcomes, COMET is limited by the occurrence of persistent epithelial defects. Another non-limbal approach utilizes natural multipotent mesenchymal stem cells or cultivated iPSCs. In 2016, human-induced pluripotent stem cells were cultivated on a maintenance medium to form two-dimensional organoids called seam, or the self-formed ectodermal autonomous multizone as seen in the picture. From them, the corneal epithelial sheaths were obtained. Emerging tissue engineering strategies aim to enhance stem cell proliferation. They're limited to animal studies and include the inhibition of nedylation and the upregulation of anti-apoptotic WNT beta-catenin pathway. Though not as dynamic as the epithelial cells, the role of stromal stem cells are being explored for corneal scars. Interestingly, allogenic endothelial stem cells are the only approved regenerative therapy for bullous keratopathy, marketed as Visnova in Japan. An iPSC-derived endothelial substitute is currently in the pipeline. In the retina, stem cell therapy remains valuable for neuroprotection and cellular replacement. The retinal pigment epithelium is usually the primary target since it does not require any synaptic reconstruction. With poor visual outcomes, RPE replacement has been tried for Stargardt's disease with embryonic stem cells and age-related macular degeneration with induced pluripotent stem cells. Unlike the RPE transplantation, the other retinal transplants don't fare too well. One such method is the transplantation of photoreceptor progenitor cells from embryonic stem cells. But current research is now focusing on unlocking the healing mode of Muller glia cells. Two-dimensional structures formed from induced pluripotent stem cells simulating the retina are called retinal organoids. These are being tried for retinal degenerations. As the retina and the visual pathway lack resident stem cells, most of their degenerations are managed with gene therapy. The first approved gene therapy worldwide was Luxturna, developed for Leber's congenital amaurosis. Basically, here, a functional copy of the RPE65 gene was delivered using an adeno-associated viral vector 2 into the subretinal space. This virus, in turn, hijacked the cellular machinery to produce vital photoreceptor proteins. Major clinical trials showed improved visual function in around 69% of the patients, though more than half experienced IOP spikes. A childhood variant of this blinding disease caused by centrosomal protein CEP290 mutations is tackled by the CRISPR gene editing technology. Basically here, molecular scissors or Cas9 create nicks at regular intervals within the genome, which is filled by a programmed DNA. This system is also introduced subretinally using a different viral vector. The results were promising despite few patients having vision loss. Retinitis pigmentosa patients with these mutations were also included in both these trials. While understudied, stem cells and their markers in the rest of the ocular and periocular tissues hold great promise for future therapies. They are being tried for the management of orbital reconstruction, ocular surface disorders, myopia, anterior uveitis, ciliary body detachment, open angle glaucoma, and cataractogenesis. To sum up, regenerative ophthalmology is a rapidly evolving field employing stem cells, gene therapy, and tissue engineering. From limbal techniques like the CLET, CLOW, and SLET to non-limbal procedures such as COMET, their focus is mainly on the cornea, especially the epithelium. Retinal regeneration, particularly in cases of Leber's congenital amaurosis, is primarily driven by gene therapies like Luxturna and the CRISPR gene editing. The role of stem cells in the rest of the anterior segment and periocular tissues are being researched upon in animals. This article unfortunately did not cover advances in neuroophthalmic regenerations, which will be briefly touched upon by my mentor. Thank you.
So, Dr. Tinku is our member ARC North, and she is the coordinator for today's program. Can I ask her to continue with the program?
Hello, good evening. So that was a wonderful journal article and very well presented, and highlighted all the points very well. So I would request Dr. Namrata Sharma ma'am, please give her comments on this article.
So I think one, I would like to congratulate Dr. Prashant Bhavan for doing this journal club for the postgraduates, which is often not covered in many medical colleges. You know, we do case presentations, we do so many things, but journal club is often left. And thank you, Dr. Tinku Bali, for coordinating this. I think it was a very well presented article, which was done by Dr. Rohan Basak, and it covered almost everything. I mean, initially I thought it is only going to cover cornea, but it also covered retina. Now, as far as the status of the stem cell in ophthalmology is concerned, although if you look at the regulatory bodies from ICMR, that is Indian Council of Medical Research, stem cells of all the other systemic disciplines in medicine, like, endocrinal, like neurology, like everything has come as the standard of care. But unfortunately, it is only the ophthalmology stem cells which are still not considered as the standard of care, which, as ophthalmologists, we all are actually fighting for. But as far as the postgraduates are concerned, I think very well covered to say that CLAU is a procedure for cornea, and so is the SLET, which came from India by Dr. Sangwan. Then the mini SLET, which came for DUCLET, is cultivated limbal epithelial transplantation, where you require a lab, and where you require sometimes, because you have to cultivate those cells in the lab, and it is very expensive to have a lab. And so, like, growing stem cells in the lab vis-a-vis growing stem cells on the eye. On the eye is something like SLET, which actually was explained very well by Rohan also. So SLET has become this, you can say, unofficially the standard of care for all of us. And we like to do it, but when there is conjunctival involvement, which is quite a lot, then we like to combine it with CLAU also. COMET is there, and COMET is mainly done where, you have bilateral involvement, and you cannot take the limbal stem cells from the fellow eye and also from the relatives. So COMET, again, he alluded to in the study. Also the mesenchymal and the adipose stem cells, because they can sort of convert into any kind of cells or any kind of origin they can have. So a lot of work has gone into it, although clinically I don't think it has made too much of a difference, although in lab there is a lot of work which is going on it, and people are hoping that, mesenchymal stem cells, if you put, then maybe they take they become like the originator cells, or like the primitive cells. So I think as far as the cornea part is concerned, we are doing it, but I wouldn't say that in that way we've reached there where we had to. Only one, Dr. Kinoshita's group, has been able to commercialize it, the stem cells in that way. And I mean, rest of the world is actually following him. And I think that is it from as far as the stem cells in anterior segment are concerned. I think Shweta can add more to it, because Shweta is also working on it.
Thank you so much, ma'am. That was a very comprehensive, overview you gave of all these new regenerative therapies. So yeah, so I now invite Dr. Shweta to give us her viewpoint on the journal article, as well as, add something, give her perspectives on this.
Thank you so much, Dr. Tinku. And I think Dr. Namrata has very beautifully covered everything to what Rohan already presented. Again, a great presentation, Rohan. Congratulations for that. I really have very few points to add to what Dr. Namrata has said, because she covered most of it. In patients where you have a unilateral disease, the donor is usually the other eye, which is what we call as the autologous, which is where the SLET or the auto-SLET comes in, or the CLAU comes in, which is conjunctival limbal autograft. However, like a COMET is an alternative when both your eyes are involved, where you're using the oral mucosa. Similarly, when you have a bilateral involvement, there is something called as an alloslet, where a cadaveric donor cornea is used to facilitate or to provide the limbal stem cells in patients who have a bilateral limbal stem cell deficiency. But in such cases, a long-term or a lifelong oral immunosuppressive is something that is mandatory to keep those cells alive, because it's a highly vascular structure area where you're going to be putting you would expect the limbal stem cells to go and settle. Well, we really do not have any documented evidence to say where these stem cells migrate, but we do believe that they go and settle in the niche, because even after you have done the slet or a stem cell transplant, and the patient undergoes a corneal procedure like a DALC, a lamellar keratoplasty, or a full thickness corneal transplant, these patients do show evidence of the stem cells which has been transplanted, and rarely do they have a persistent epithelial defect or thing. So probably they do migrate, but we really do not have fixed answers to it. As far as the cadaveric stem cells are concerned, in addition to the chronic stage for providing vision, they are also often commonly used in the acute stage of chemical injury, where you have severe chemical injury and the entire surface is raw. So to facilitate faster epithelialization, alloslet has been used in the acute stage of the injury to facilitate epithelialization, because it's cadaveric donor cornea, so you're not really worried about the amount of tissue that needs to be harvested. But other than that, I think it broadly covers everything. As far as slet is concerned, because it's in the eye, like Dr. Namrata and Dr. Rohan specified, the expense is very minimal, because you do not need a GMP certified lab. But otherwise, to grow cells, which has been the way to culture stem cells in Europe, you would need a GMP certified lab, which is a huge financial burden. So, I think anything more?
Thank you, Dr. Shweta. Yeah. So those were some very important points that you highlighted. And I'd like to invite Dr. Parvez Khan from GSVM Medical College, sir, for your perspectives on this.
Thank you, ma'am. First of all, I'm really happy that people in India are now discussing stem cell, because a few years back, when I was into this, I was basically was into stem cell implantation for retinal diseases, especially retinitis pigmentosa and AMD and rods and Cons disease, Stargardt disease. The problem with stem cell in India is this, that there are a lot of regulations, which is not very clear cut, how to proceed with it. Even the stem cell, there's a committee in the stem committee has been constituted by the government of India. But when you go out to take a permission, that it becomes really difficult. It keeps on lingering for years together. And if you are doing it without that regulation, then automatically some type of legal problem arises. Still, we have been able to do around 20-25 patients, and the result was very encouraging. I'm talking about Wharton jelly derived mesenchymal stem cell, and with the stem cell, there's an issue that the stem cell, as we all know, that stem cell has to, if you want to regenerate RP, if you want to regenerate the photoreceptor, the stem cell has to be implanted in that area. So in India, there's a company called iStem. They have already doing a clinical trials in RP center and LVPI. They are iPSC derived RPE. They are putting subretinally. But the problem with this, most of the country where the clinical trials have been occurring, they are implanting subretinal. Subretinal requires a lot of surgery, and it's a cumbersome procedure with fraughts of endophthalmitis or detachments or hemorrhage. Now, going after all this research, what I tried was giving it suprachoroidally, because that's the closest proximity where you can enter go near to the retina without actually entering the eye. So, and the result was very encouraging. This research article, which Dr. Basak has been very exhaustive. It's gone through all the anterior segment as well as posterior segment. I think the future of all this non-curable disease lies in stem cell. We have to, one way or the other, we have to go through this process. And I think the future lies with stem cell.
Sure. Very rightly pointed out, Dr. Parvez, sir. So with that, before we move on to the next article, I'd like to request Dr. Manideepa if she would like to add something to this or tell us why she chose this article.
Yeah,
Dr.
so
Manideepa.
this is actually a very extensive article, and Dr. Rohan has done a very good job in presenting. And thank you, Namrata Sharma madam and Dr. Shweta Agarwal madam, who has further elaborated on the corneal topics. I would just like to add a little bit lesser talked about topic which the journal didn't cover. That is in neuro-ophthalmology, where ocular regenerative therapy is showing a huge scope, that is Leber's hereditary optic neuropathy. So that is commonly known as LHON. So as we all know, it is a maternally inherited mitochondrial disease, and the mutations mostly are ND1, ND4, ND6, the common location being the 11778 G28 mutation. So for the PGs, I would like to highlight that gene therapy in this disease is now, in future is going to be maybe a long question or a recent advances topic. So need to thoroughly read about that. The clinical trials are the phase 3 clinical trials that are going on, mostly on the gene product, that is the recombinant mitochondrial ND4 DNA, that is the GS010 product, known as the Linado gene. And what they are doing is they are giving intravitreal injections, either unilaterally or bilaterally, in patients who have bilateral visual loss less than either 6 months or 6 months to 1 year duration. So in those patients, there has been a significant improvement in terms of initial improvement, as well as a sustained visual improvement that has lasted till the follow-up of a 5-year study that is known as RESTORE study. So that is one. And the other things, those are the gene therapy and the base editing ones. Those are still in the preclinical stages. And the other therapy, which are mutation independent, they are mostly targeting the oxidative stress on the retinal ganglion cells. And thereby it is reducing the retinal ganglion cells apoptosis. So as you can see here, that the commonly used one, which nowadays we are also using in our practice, that is Idebenone, which is a coenzyme Q10 analog, which crosses blood-brain barrier, and it restores the ATP levels in the mitochondria, which reduces the oxidative stress of the retinal ganglion cells and reduces its apoptosis, followed by a improvement in the vision. So this has been tried in one of the RODOS clinical trial, which is also a phase 3 clinical trial. And there they have given 900 milligram per day for a period of at least 12 to 24 months. And further studies, which has done a follow-up till 30 months, RODOS follow-up trial, has shown a sustained improvement in vision. But sadly, here in India, we have formulations of 45 milligram tablets. So giving a patient of a 900 milligram per day dosing is like 20 tablets in divided doses. So that is a little bit of a trouble. But then in patients whom we have used the treatment, it has shown significant improvement. So that is one breakthrough in LHON. Nowadays, we don't have to counsel the patients that there is nothing and only give vitamin supplementations and counsel them for the visual loss or give low visual aids. But nowadays, on the other hand, we are able to give some treatment and there is some light at the end of the tunnel. So other treatments, as you can see, for example, the brimonidine or cyclosporine A or the other treatments which had shown a neuroprotection to a certain extent in glaucoma, but they are not of that much use in neuro-ophthalmology from the LHON side. And gene therapy, the various targets of these adenovirus associated vectors, these are still in the preclinical levels. We hope that something again fruitful comes out in the future. So every day there is some bit of a change or something new coming up. There is no end to it. And preclinical trials are moving towards clinical trials. So let's see in the future, maybe some other promising treatment might come up in these patients where the visual loss as of now is irreversible.
Thank you, Dr. Manideepa. And I thank all the panelists and the speakers for giving such an excellent overview of the emerging therapies that we are going to be hearing about more in the future. So thank you all. Excellent presentation. With that, we move on to our second journal article. And this article will be presented by Dr. Shubham Darade, who's a final year DNB resident at the Narayan Netralaya, Bangalore. And mentoring him is none other than Dr. Rohit Shetty. So Dr. Rohit Shetty is someone who has pushed the boundaries of clinical innovation and research and is one of the most distinguished clinical scientists in our country. So it's a real pleasure to have you here with us, sir. And we look forward to the article and also your perspectives on it. So thank you so much. And with that, I invite Dr. Shubham to share his slides.
All right. Good evening, everyone. So I have the pleasure of sharing with you my insights on an article from Molecular Therapy. We're going from one end of the spectrum of evidence-based medicine, where we saw a review article, to an animal study titled Safety and Efficacy of CRISPR-Mediated Genome Ablation of VEGF-A as a Treatment for Choroidal Neovascularization in Non-Human Primate Eyes. This article was published in the Journal of Molecular Therapy, volume 32, issue 12, which has an impact factor of 12. And the authors sought the various required permissions as are indeed indicated in an animal study. So, as we all know, pathologic angiogenesis, as in retinopathy of prematurity, PDR, and neovascular AMD, is mediated largely by VEGF, DDGF, fibroblast growth factors, insulin-like growth factors, etc. Currently, VEGF-A is proven to be the most successful pharmacological target. Current approved therapies in clinical practice include anti-VEGF injections, intravitreal injections such as bevacizumab, ranibizumab, or brolucizumab, aflibercept, etc. However, these carry the risk of frequent intravitreal injections, which are invasive. They always are associated with the risk of endophthalmitis, and they cause a significant burden to patients and healthcare system overall. So the authors have looked at gene therapy approaches where they've used adeno-associated virus vectors to express these anti-VEGF agents. Now, these are done using CRISPR, which is clustered regularly interspaced short palindromic repeats, where CRISPR-associated endonucleases are used to create double-strand breaks, which cause insertion-deletion mutations to disrupt the target gene. Now, the source of this Cas9 could either have been Streptococcus pyogenes or Staph aureus. The authors have chosen Streptococcus pyogenes, and they've also used guide RNAs to target gene editing activity at the desired site. So as we can see, the VEGF-A exon protein coding region, where either gRNA1 or gRNA2 could have been used as a promoter sequence. But through in vitro studies, the authors found gRNA1 to cause a greater VEGF-A suppression. So coming to materials and methods, the aim of this study was to study safety and efficacy of CRISPR-mediated genome ablation of VEGF-A in non-human primate eyes. So to do this, a dual AAV vector design was used, where SpCas9 and gRNA were incorporated in dual AAV8 vectors, and empty gRNA was used as a control. Around 33 adult rhesus macaques were screened for native antibodies against AAV8, and 4 rhesus macaques were selected. So we had a sample size of 8 eyes of 4 rhesus macaques. In these monkeys, a subretinal injection of about 300 microliters given using 38 gauge subretinal needle, superonasal to the fovea. It included AAV8 SpCas9 and either AAV8 gRNA1 or AAV8 empty control. So these were delivered in either a low or a high viral dose. So this summarizes the regimen of imaging as employed by the researchers, where a baseline color fundus photo OCT and ARG was obtained, and post-injection at 2, 4, 6, and 8 weeks, the fundus fluorescein angiography and color fundus photo was obtained, followed by at week 12, where a necropsy was done, and histopathological as well as immunohistochemical examination was done. So all the statistics that I'll be displaying were done by these following methods, where a p-value of less than 0.05 was considered statistically significant. Coming to the results of the study, The authors did not find any significant change in intraocular pressure, and they could not find any evidence of intraocular inflammation in terms of anterior chamber cells or vitreous haze at any of the weeks. So this image shows, it's a representative color fundus photo, scanning laser ophthalmoscope image, and an OCT as captured at baseline, week 2, week 8, and week 12, after giving a low viral dose of AAV8 SP-Cas9 with control and with guide RNA. So at week 2 is when the AAV, the subretinal injection was given, and at week 8 at week 4 is when the laser-induced CNV was obtained. So as we can see on the image, the cross points at the mark of the at the mark where the injection was given, and the dotted line shows where the 300 microliters of injection has spread. Interestingly, in this, two interesting findings were concentric rings of hypo and hyperreflectivity on the SLO, outer retinal disruption as marked by these arrows, and a subfoveal deposit, as we'll be discussing further. Similarly, when we observed a similar response after injection of high vector dose, however, we saw that the retinal disruption was more prominent in eyes that received this higher dose. Again, a fundus fluorescein angiography was also performed, where on the left-hand side was a grading system used by the researchers, where each of the 8 laser-induced CNV sites were graded from grade 1 to 4, and an average was obtained. This showed that eyes that received a low dose of AAV8 SP-Cas9 and gRNA showed lesser CNV leakage compared to those with empty gRNA controls. However, when we looked at the higher dose AAV8, higher dose viral vector, no significant difference was found, which could be attributed to the extensive outer retinal disruption that was caused. At necropsy, these are the representative fluorescence confocal microscopy images, where we can see the green fluorescent protein hemagglutinin tag, RPE65, as easily evidenced in this, where the VEGF protein concentrations as measured from the macular tissue using ELISA showed a 33.3% reduction in VEGF levels in eyes that received an active gRNA1 compared with empty gRNA control, but only at a lower viral dose. At a higher viral dose, the overall VEGF-A levels were extremely low. Now, at 12 weeks after the subretinal AAV8 SP-Cas9 injection, they also observed rod and cone disruption characterized by loss of photoreceptor nuclei in the outer nuclear layer and disruption of inner and outer segment morphology, which was seen at both doses, but again, more severe disruption at a higher viral dose. The inner retinal layers also showed an increased GFAP expression in astrocytes within the retinal nerve fiber layer. And there was also loss of ramified morphology of the resting microglia. These findings suggest that subretinal AAV8 delivery of CRISPR-Cas9 may lead to local photoreceptor disruption with glial and microglial responses. To further explore the identity of the subfoveal hyperreflective material as observed on OCT, at necropsy, an HNE staining and immunohistochemistry was performed, where these accumulations were mainly found to contain RPE cells, which were tagged with GFP HA tag, which again shows that this subretinal fibrosis is indeed composed of RPE cells which are co-transduced by both SP-Cas9 and gRNA. The presence of RPE, collagen, vimentin within this material is suggestive of the subretinal fibrosis. And again, it was consistent with the fundus appearance and imaging characteristics of this material. The authors also looked at the immune response, where initially no clinical signs of intraocular inflammation were noted. However, there was a raised aqueous CCL2 at 1 month and 3 months. Again, none of the other chemokines, such as any of the interleukins, interferon gamma, TNF alpha, or even VEGF, none of these were elevated in the aqueous. And no immune response was detected to either the viral capsid or the transgene in blood and aqueous. Which leads us to the conclusion that the local inflammatory response in the form of subretinal fibrosis was likely due to the retinal damage rather than host immune response to the vector or transgene. So in conclusion, there was a 33% reduction in VEGF-A production as per the study. An unexpected finding that the authors found was acute outer retinal disruption characterized by subfoveal fibrosis photoreceptor damage, and retinal inflammation. And this is something that needs to be studied further. There was no host immune response to either the viral capsid or the transgenes, which further solidifies the immune privileged nature of the subretinal space. And unlike conventional gene augmentation therapies, which show diminishing efficacy over time, gene editing directly modifies the host genome to enable permanent corrections of disease-causing mutations and potentially long-lasting treatment effects. Unlike other studies, this study was not without its limitations. There was a minor flaw in the design of the experiment, where the timing of CNV induction in relation to the subretinal injection does not really simulate the real-world scenario. The study had a very small sample size, probably due to the high cost associated with non-human primate studies. There was absence of additional control comparison groups. There was a limited follow-up, and at 12 weeks, there was actually no significant reduction in clinical CNV grade. Future studies employing different AAV vectors, a lower vector dose, or cell-specific promoters may be necessary to determine the safety of AAV-mediated CRISPR platforms. These were the references by the authors. Before I conclude, I just wanted to highlight some of the gene editing gene therapy development at GrowLab Narayan Nethralaya Foundation, where we have collaborated with IGIB New Delhi in emerging opportunities for gene editing therapies. Where this is a very interesting video, which kind of shows what they talked about in the animal study as well. So live cell imaging in human epithelial cells with the AAV vector gene therapy. This shows expression of the green fluorescent protein, which lights up the cell. And therapeutic proteins can also be expressed in a similar way to help treat patients with many types of diseases. Another area of work where GrowLab has worked in is in developing a gene therapy for FENID, which is a very rare neurodegenerative disease, familial encephalopathy with neuroserpin inclusion bodies. And this is the article that was published, Hope, Despair, and CRISPR, The Race to Save Women's Life, which is actually a tragic story of one of the patients who suffered from this. All right. So I would like to thank Dr. Rohit Shetty, sir, and AIOS and ARC for giving me this opportunity, and also the authors of the article for sharing the article with us. Thank you.
Thank you, Dr. Shubham, for so well highlighting a potential therapeutic option, for VEGF suppression. So I think it's ideas like this that would take science forward. And right now you said that it does have its limitation. There was so much of fibrosis, but the anti-VEGF suppression, as shown in this study, is something that could be taken forward. So I'd now request Dr. Rohit Shetty, sir, to give his perspectives on this article and tell us a bit more about this field and why you chose this article. Dr. Rohit Shetty, sir?
Yeah, thank you, ma'am. And thanks, ARC, Dr. Namrata, for creating such a wonderful platform for these kind of discussion. This is how you bring in new concepts to the mind. Because if you look at next 10 years, most of the disease is going to be looked at it from genes or regeneration, what was discussed by Dr. Rohan. Both the young speakers very nicely brought in these concepts. There are a lot of discussion about CRISPR and Cas9. It's important for ophthalmologists and all the youngsters out here to understand what is CRISPR, because see, we in our lab use it very routinely, and I'm sure a lot of people, scientists, are going to use this in their practice. It's more like an editing software. But if you look back at the history of how it was discovered by chance. Yogurt, which was there in part of US, there were scientists, that's what they say, science is all about keen observation. It's not about intelligent mind or having lots of funding. Many places which has lots of fundings don't do any great research. It's a fire in the belly, and no fund is what brings in revolution. So what happened was they were observing that the yogurt, which had infested with this, what he said, Streptococcus and there are so many other Streptococcus bacteria, they've somehow had a beautiful way of preventing virus from coming into the to the yogurt. So some smart scientists were in the lab asked what makes the bacteria so so prevalent here and not the virus. So what they did was they took out the bacteria and they started doing the DNA of the bacteria. Interestingly, when they looked at the DNA of the bacteria, they found that the bacteria's DNA had a viral DNA in it. That means the bacteria has taken a viral part of a DNA into its DNA. The question came is why does a bacteria has to have a viral DNA in it? What makes the bacteria so smart? So then the bacteria was very smart. What it did was when it exposed to a particular virus, it would take the viral DNA in it and have a copy of it ready. And that copy is what you call it as the CRISPR. Clustered, regular, interspaced, short palindromic, repeated sequence. That is the name for that, the whole thing when we call it CRISPR. It's about how those clustered particles were all organized by the bacteria. And what's interesting is if the same bacteria was exposed to the same virus, what it would do is Cas9, which both the speakers spoke about, is an enzyme. It's like a scissor. So what it would do is it knows exactly at what viral gene it has to cut. So it would send the same Cas9 to that virus and it would cut off so that the virus becomes ineffective. So smart the nature is. So the scientists who have studied this understood the bacteria had this gene which would take up the virus and take the virus into its own thing and cause this CRISPR. Because it was clustered, because there was the sequence, it was interspread, it was short, the sequence was very short, and it was repeated. That's why CRISPR. And Cas9 is just an enzyme. So what they did was, if you take the same Cas9 and bring it, it becomes your gene scissors. It's an enzyme. So you can cut any part of DNA, it can go into it. And that became what the world today talks about the editing and all that. Like I said, all these articles bring in a very fresh outlook. And here I think they're looked at from the VEGF point of view because there are specific agents which can block it. But the one he spoke about, that girl, which we were featured in Nature journal, is there was this girl who had a very specific mutation. And what is interesting here is in gene therapy is that mutations now, like for example, Luxturna. Luxturna needs a specific gene mutation for it to act. Otherwise, it's not a generalized one. So, and only if you have an RP65 or one of those mutation, it will act. But what gene therapy we want to look forward is it can be specific to an individual gene abnormality. For example, this girl had a specific gene and we were running against time because she had a very short time to survive. And we collaborated with a team in Delhi and we created a gene, we created a vector. We were just a month away from injecting into her. It worked in animals. It worked. We created the same gene defect in an animal and we it worked. And unfortunately, that would have been the world's first customized gene to a patient. And she was just passed away just few weeks before we got all the permission. The permission came from PMO's office because they wanted it to be done. So I'm saying that it's not just about Luxturna or any product. It's about tomorrow. It's about customizing. For example, one approach we are following in cornea, which we have finished all the regulatory one. We're waiting for the final one is on the patient. It will be the world's first gene therapy on a cornea. We call it a gene augmentation. That means we are not changing any gene. We're not cutting with the Cas9. We are not editing anything. the gene will go and only put efforts on the genetic factors which is not functional. For example, LOX, which we are targeting. Lysyl oxidase is necessary to keep your collagen tighter. In keratoconus and many diseases, the collagen is loose. So what it does, it doesn't alter the gene. So I don't need to check for genetics at all in this patient. I don't need to look for the gene. So gene augmentation is going to be the next big change, because you don't need to worry about whether you have RP65 or whether you have ABC4, whatever gene. It will work on anything which will augment what is not functional. So that means your approach becomes mass approach. You don't need to worry about, are you having this effect? And the second approach is what we were trying to do with this girl, which we were unsuccessful because the destiny was much more stronger than us, is to create a specific gene therapy work for particular diseases. So I think it's quite exciting field. Like I said, it's not like he said in his limitations. There are challenges. Some genes don't work. Some things cause fibrosis. Something do the opposite. And when we started the gene work for keratoconus 7 years back, when I injected the genes into the cornea, all my rabbits died. Then we were wondering what happened to these rabbits. Then we realized too much of lysyl oxidase can cause the heart to freeze. All of them died of heart attack because systemically we didn't know the dose properly. I'm just making it very simple, but the challenges, like Dr. Parvez sir said, it's a big challenge. But like I said, this is what we have to do. This is what we have to work on. This all the youngsters. Nothing comes easy. I'm sure all Dr. Namrata ma'am, Dr. Sangwan, and Dr. Shweta saw a beautiful presentation of us in Goa. And it's anything which comes easy is not worth it, because it will come and go. But for all the youngsters, it will be very, very interesting if you take this up in your fellowship or your post-graduation, because the next generation of ophthalmologists should be aware of all this. It's not just for Journal Club, it has to be for your future. Thanks, Dr. Tinku, for giving this opportunity. And I think Dr. Shubham did a great job. It's a very complex topic, very complex paper. He spent a lot of time understanding with the scientists, and I'm sure he did it with a lot of understanding than just presenting it. And that's what I think every fellow or even the previous presenter was excellent. Thank you.
Thank you. Thank you, sir. Quite fascinating to hear about all these things from you. And as you rightly said, this is the future. So we all have to be updated about this. And thanks to you, you introduced the topic so well. I'm sure it has, it'll prompt a lot of youngsters to go and read about these things and get updated. So thank you so much. With that, I'd like to request Dr. Parvez for his perspectives or inputs on the article. So anything that you'd like to add?
First of all, Dr. Shetty has so oversimplified the things. Even a person with no knowledge of gene therapy and all was finding it so interesting. The very concept of gene augmentation has a new perspective, because now, as we know, Luxturna was there for a long time now. But its use has been limited because it's acting only on particular genes, RP65. Whereas we know that RP has around 240 gene mutations. Effectively, Luxturna becomes not very useful as far as the disease is concerned, because it's so limited with this sir's new this gene augmentation. I think the future is there. There are a few things I'd like to ask. How do the results compare to the standard anti-VEGF therapy in terms of efficacy and frequency of the treatment? This VEGF knockdown, VEGF-A knockdown. Just if the presenter can elaborate on that.
Yes. So in this study, there was only a 12-week follow-up. So even though there was a 33.3% reduction in the study, 12 weeks is not long enough to make a comment on whether it is going to cause long-term VEGF suppression. Another thing, clinically, when we give anti-VEGFs for these neovascularization diseases, we don't actually measure the VEGF as they did in the study. So here, the VEGF was measured after necropsy from the macular tissue itself. Now, that sort of VEGF concentration we will never be able to measure in an actual human eye.
And there was something else also. This is no long-term safety. We have not seen the long-term safety of
We have
this
not seen
permanent.
that. No, sir. No, sir.
Yeah. Okay. Thank you.
So, Dr. Manideepa, anything that you would like to add to this?
No, not really.
Okay. So
I'm
think
really amazed at how Rohit Shetty sir has brought the story of CRISPR
True.
and explained everything so nicely that I have nothing more to add. Thanks a lot for this opportunity to everybody, and very happy to be a part of it and to listen to such enriching things. So
Thank you.
thank you all.
Just to, because it's so important, because we all are, a lot of us are, we keep talking about it and we go and give talks about CRISPR, this, that, and everything. But at some point of time, it's very important for us to know, because sometimes patients may come and say, what is CRISPR? What does it do? I mean, in the beginning, I thought it's a name coined by something, but it's not. It's got a very fancy, big, So it's very important for us, because it's because people are going to be more smarter than us, because with AI and all this help, patients are going to come and just ask you about it, and it's going to be tough. I mean, just for all the youngsters out here, just be very good at whatever you know, because patients are going to be tough and more smarter than you.
True. So thank you. With that, I think we've come to the end of our time. And Dr. Rohit, it was so fascinating to listen to you. You gave such wonderful insights and perspectives on the topics. So thank you so much. I also thank all the other panelists, Dr. Parvez, Dr. Namrata Sharma ma'am, Dr. Shweta Agarwal, for their presence. Also, a pat on the back to the young presenters, Dr. Rohan Basak and Dr. Shubham Darade, for having presented such complex articles, for having dwelled so deep and done so much research to get all that information out. So hats off to you, did a wonderful job, both of you. And all this was, I'm sure, something very new for a lot of people. But, all the insights that were shared by the panelists has, started journey, I think, for a lot of people. You simplified the CRISPR technology so well, Dr. Rohit Shetty. So thank you so much. Wonderful knowledge was shared in this episode. And with that, I also thank Dr. Manideepa Banerjee for her presence. And thank you so much. Looking forward to more such, episodes in future. Thank you. Thank you, everyone.